Hello, everyone, and welcome back. Sorry I cut that last video off. It's because we just did this past slide, and this was just the other function of complement. And then now we start with the second part of the lecture. And so now we're going to talk about the innate immune system still, but now we're going to talk about another component of it, and these are going to be the professional phagocytes. Okay. So, what are the professional phagocytes? These cells are cells that um, um, they do phagocytosis. They eat other things, right? And so a big example of the macrophage, which just means big eater. And even as far as cell size goes, it's relatively large if you look at it at a histological slide. Um, so every body surface with exposure to the outside world has a big population of macrophages. This is going to be like your alimentary canal, like your GI system, your pulmonary tract, your skin. Also, it's going to have a lot of macrophages because, as you can imagine, we're in contact with like debris and bacteria, viruses, and whatnot on the daily. So we have this big population of eaters who are constantly on guard, right? Now, the life cycle of a macrophage is a bit interesting because macrophage actually doesn't begin its life as a macrophage. It actually starts as a monocyte, which is a cell that goes in the bloodstream, and then it actually take up residence in a tissue and then become more adept to living a tissue life rather than a circulatory life. And then it becomes a macrophage. Now, under different circumstances, a monocyte can actually become a dendritic cell, but we'll talk about that in a later lecture. So the thing about macrophage is that they have three moods, and so you can think of it as like a resting mood. And so this is a very dormant stage. Basically, the macrophage is just picking up debris from the surface where it's stationed, dead cells, dust, really small things, very dormant. And also because it's not dealing with a lot of antigen presentation, because it's a very dormant phase, to present the T cells, it doesn't have that much MHC class 2 expression. However, upon having some danger signals, which we'll talk about later, a macrophage can enter a primed state, which means it has more MHC class 2 expression. That means it can show more antigens to T cells and possibly activate the adaptive immune system. And mind you, it'll also put up the co-stimulatory factor B7, which binds to T cell CD28, but beside the point. And also in the prime state, the macrophage has higher phagocytic activity. Then there's even a hyperactivated state, and it's the highest phagocytic activity. The cell even gets bigger and scarier, and then even produces something called tumor necrosis factor. Specifically, I believe tumor necrosis factor alpha, but that's beside the point. And we'll talk about what this means in a second. So to enter from the resting state, which is the default state, to the prime state, the macrophage has to be woken up essentially. So this can be done different ways. Helper T cells and natural killer cells who already know there's an infection going on can wake up the macrophage by pelting it with something called interferon gamma. And I think like gamma is G for good morning. You're waking up the macrophage in a sense. Once the macrophage wakes up, it'll upregulate its MHC class 2 presentation, and it becomes better, antigen presenting to further activate other T cells. And so forth. Then there's even hyperactivated state. So what distinguishes hyperactivated from primed? Well, if the primed macrophage detects a red flag, it becomes hyper alert and aggressive. And so what is that red flag? Usually this will come in the form of a molecular sequence pattern type thing that the body has evolutionary grown to recognize as dangerous. So for example, gram-negative bacteria have their lipopolysaccharide layer. We have specific toll-like receptors in the body, pattern recognition receptors in the body, through evolution have learned that LPS is a red flag. If that's in the body, you definitely have a gram-negative and you should start panicking. If the macrophage's specific receptors pick on say that, or like mannose is something else, another red flag. the macrophage enters like its very aggressive phase. And so when it's in a very aggressive hyperactivated state, the macrophage increases its size, becomes more phagocytic, and produces a cytokine called tumor necrosis factor and interleukin-1. Tumor necrosis factor is a chemical that the macrophage will pelt virus-infected cells with the virus-infected cells with, and also it's also called tumor necrosis factor because in addition to virus-infected cells, it also kills cancer cells, hence the name tumor necrosis, necrosis being death. Now, the specific mechanism of TNF is variable depending on where it's used, but in the case of cancer cells, it's actually believed to target the vasculature supplying the blood to the tumor, and so therefore it's believed that tumor necrosis factor alpha binds the blood vessels going into the cancer cell, essentially ligates them, cuts them off, and then this promotes the tumor cell kind of starving to death from a lack of perfusion, blood supply, right? But that's aside the point. And then as for IL-1, interleukin-1, think of it as the first flame. That's the mnemonic I use. It's a pyrogen. That means it triggers inflammation, heat, and so when you have heat, that's it facilitates a lot of immune function. Inflammation makes makes a lot of vasodilation, makes it easier for other cells to like enter that site of infection in some cases, things of this nature. And so again, this is the more specifics I was talking about before. How do the macrophages detect the red flags? These are through pattern recognition receptors. These are receptors that have evolutionarily grown accustomed to detecting red flag patterns. And these red flag patterns are called damage-associated molecular patterns. So PAMPs detect DAMPs, PRRs detect PAMPs, and a specific type of PAMP is a DAMP. It's not too important, just more so you understand the concept. And so I think it's important to know these because I think they will come up in other subject matters. There's different types of pattern recognition receptors. And a specific type of the toll-like receptor, which I alluded to earlier. And there's different subtypes of toll-like receptors that detect specific, molecular patterns that are historically considered dangerous. So TLR4, for example, detects the LPS layer of gram-negative bacteria. TLR7, single-stranded RNA virus. TLR9, double-stranded DNA virus. And mind you, the toll-like receptor doesn't have to be on the macrophage's outside membrane. It could actually be useful to have these receptors inside the cell, like on a phagolysosome, because viruses tend to be inside cells. So having receptors inside your cell can actually be beneficial in this regard. Next cell we have in innate immune system is called the neutrophil. It's very short-lived. It's like the foot soldier of your body. 75% of our white blood cells are neutrophils. They are not antigen presenters, though. They're actually in a class of white blood cells called granulocytes, and they have sisters called eosinophil and basophil, which we'll talk about later, and then also at a much later lecture as well when we talk about allergy. But yes, neutrophils are highly phagocytic, so they can eat and then they destroy. So they take up and then they like self-destruct with whatever's damaging inside of them. They also produce tumor necrosis factor. So it's almost like a cute little macrophage. But also it's called the whole class, neutrophil, eosinophil, basophil, is called granulocyte because they have granules, and the granules basically just a little pocket of highly dangerous chemicals inside the cell granule. And these are not only are they harmful to the pathogen, they're actually harmful to our own cells, right? So it's kind of like a collateral damage type deal. Also, neutrophils, this is more recently studied, they can form something called nets, neutrophil extracellular trap. They basically just kind of like cast themselves, like, the casting that when you catch like bait fish when you fish, if you fish, I fish. They form like this web, they kind of cast themselves, and that's another way of like catching invaders, right? And mind you, like when you squeeze a pimple, or you squeeze someone else's pimple, or you watch someone squeezing a pimple, or you watch someone squeezing your pimple, or anything of that nature, the white pus that oozes out is actually white blood cells, and most of those white blood cells are actually going to be neutrophils. And mind you, there's mainly neutrophils because most blood cells, the foot soldier who does the dirty work and a lot of killing, is a neutrophil. So the neutrophil actually has sounds very violent, but it's actually rather sophisticated in how it gets from point A to point B. So let's orient ourselves. Like most blood cells, hence the name white blood cell, right? Blood cell. The neutrophils in the blood. And so then you have here like endothelium, like the inside the lining the blood vessel, right? Right. So this is what we have. So this is endothelium, and you just have like a current of blood, like just whooshing by, right? Whoosh. Okay. The endothelial cells have surface markers. And so one good to know is called ICAM. So just there's always ICAM, I guess, like on the endothelium, right? So I'll just underline this orange because I have this orange here. Once stressed out, however, in other words, they know that there's a problem going on. So let's just say, here, I do like this squiggly arrow. It's like an attack. The endothelium gets stressed out, the poor little thing. It's actually kind of cute when it's stressed out. It shows the world that it's stressed out. It's very like outwardly in its emotions. It's going to project something called L-selectin, and I'll do L-selectin at this right. And so what can cause the stress? It can be tumor necrosis factor, interleukin-1, which is pyrogenic, all the things I was saying. So stressed out endothelium has L-selectin, and then endothelium at all times is ICAM, right? So now we have a stressed out blood vessel who just picked up on some tumor necrosis factor, interleukin-1, gets stressed out and is now projecting L-selectin. This is good. The neutrophil has a some receptors, if you will, or ligand. Ligand is complementary to the receptor. It's called SLIG, selectin ligand, or L-selectin ligand specifically. Neutrophil will bind the L-selectin. Okay. Which is this. I don't think I'll draw it in. So selectin ligand binds L-selectin on a stressed out cell. That's good. However, SLIG to L-selectin interactions are weak, and so it'll only cause the neutrophil to roll on the surface rather than catch it, right? So think of it kind of like Velcro. Okay, so the way mnemonic is selectin SLIG slows, right? But the story doesn't end here because we didn't talk about ICAM yet, did we? So now you have a neutrophil that's rolling on the endothelial surface, mediated by SLIG to L-selectin. But this is the neutrophil still moving. You need the neutrophil to stay still so it can dock in a place so that it can move into between the endothelial cells to get to the site of infection, which is outside the blood, right? What caused the endothelial cell to get stressed to begin with? Something outside, right? Therefore, there's a smelly region of the endothelium, which is the site of the specifically parts of endothelium that are near the infection site. So you can imagine there's a lot of like chemoattractants in this area, right? Like C3A, C5A. Remember the anaphytoxins? In the presence of the smelly region, the neutrophil is going to grow something called integrin, right? And so integrin will bind ICAM. Remember ICAM from before? So remember, we have SLIG to L-selectin. Now you have ICAM, integrin to ICAM. Upon binding ICAM, ICAM is basically like the key that the neutrophil needs to be able to do some extravasation between the endothelial cells to get outside of the blood to the site of the infection. Okay, so that's going to look something like this. So you have two endothelial cells, right? And then you can have like this neutrophil. Let's do it like blue. It can like, have you seen ocean documentaries how octopi can like squeeze in tiny places? Kind of like that. And so this is the neutrophil. It's going to squeeze through these blood cells, and this squeezing is going to be mediated by it's going to be mediated by the binding of integrin to ICAM. Okay, and that's extravasation. And so that's how the neutrophil goes from rolling adhesion into being able to extravasate through the between endothelial cells. And mind you, other cells have similar mechanisms in order to get out of the blood into the tissue. This is a general mechanism. We just use neutrophil as an example. And so again, you have other granulocytes. You have eosinophil. They release. They're also so they also have granules, and they also dump out these granules. Can be very vasoactive and whatnot. Eosinophils are typically seen elevated in allergies, and also when you're fighting a parasitic infection like a helminthic worm. You have basophils. They're very similar to mast cells, which we didn't talk about yet, but the mast cells are residents in the tissue. Basophils are in the circulation. Basophils release histamine, and histamine is really what mediates a lot of allergic reactions sometimes. But we'll talk more about these later. But just know that this is neutrophils' cousins, fellow granulocytes. Okay, and so we'll pick up here next time. Thank you.